DNA replication! Woo!
Yay!
In DNA replication, you first need nucleotides. There are two types, RNA nucleotides and DNA nucleotides. Remember, there are 5 types of nitrogenous bases: adenine, cytosine, guanine, thymine, which is specific to DNA, and uracil, which is specific to RNA. Nucleotides have 3 parts: a phosphate group, a pentose sugar, and nitrogenous bases. In DNA replication, you have DNA strands. These strands run anti-parallel, meaning that they're opposites. This is marked by the 5' ends and the 3' ends. First, the enzyme helicase breaks down hydrogen bonds between the 2 parental strands. This creates what's called a replication fork. Single-stranded binding proteins help stabilize the unwound DNA and keep it from rebinding. The enzyme primase comes next and adds on RNA nucleotides. The enzyme DNA polymerase III begins to add nucleotides to only the 3' end of the strand. This forms phosphodiester bonds between nucleotides. The enzyme DNA polymerase I replaces RNA nucleotides in the primer with DNA nucleotides. However, it does not form phosphodiester bonds. Next, the enzyme ligase comes to form those phosphodiester bonds. Remember, when a DNA strand is being made, it is always made from the 5' end to the 3' end. And that's the leading strand. Next, the lagging strand.
The first step in the lagging strand is very similar to in the leading strand. First, primase comes in and makes a RNA primer on the lagging strand. And this moves just like the leading strand, from the 5' direction to the 3' direction. Next, we need DNA polymerase III. This synthesizes new DNA nucleotides, moving from the 5' end to the 3' end. In the lagging strand, this means it's moving away from the replication fork. Since in the lagging strand, it's moving away from the replication fork, this creates fragments called Okazaki fragments. So this, you can see here, is one Okazaki fragment. So next, just like in the leading strand, we need DNA polymerase I to come in and replace those RNA nucleotides. And it replaces them with new DNA nucleotides. But just like in the leading strand, the nucleotides replaced by DNA polymerase I are not connected by phosphodiester bonds. So we receive DNA polymerase I creating new DNA nucleotides. So next is ligase again. Ligase comes through and makes those phosphodiester bonds on the DNA nucleotides that were just replaced by DNA polymerase I. Now we have one complete Okazaki fragment, and then ligase also connects Okazaki fragments together. And that's the lagging strand. Thanks for watching.